Welcome everybody. In this video we're going to talk about how well do decision support tools, or DSTs, adhere to the reporting guidelines, recommendations, or standards that we talked about in a previous video. And I've re-put these standards here, which you can go view that other video. And the first 5 again were about naming sequence variants, and then the second 5 were about test reporting itself. So to do this, I'm going to talk about a study done by Boostman and colleagues. You can see the title here along with its citation. Go ahead and look up this study. It's a good study. And so we're going to talk about this study because what they did is they looked at a total of 20 different commercial panels and essentially evaluated how well do they adhere to each one of these standards, which would be 10 in total. So let's take a look at how well these panels adhere to that. And so what we're going to write down here is the percentage that fully fulfilled each individual guideline or standard, and how many partially fulfilled it. And I'll explain a little bit more about that on the way. So on the left side of each individual standard or recommendation, we'll put the percent of those 20 that fully fulfilled it and the percent that at least partially fulfilled it, which would include the fully fulfilled. So for the first one, using Hugo Gene Nomenclature Committee to specify the gene, a total of 50% fully fulfilled it, meaning that all gene symbols in their decision support tool complied with Human Genome Nomenclature Committee specifications. However, 100% at least partially fulfilled it or more, meaning that at least greater than one gene symbol, so a pretty low bar there, complied with the Human Gene Nomenclature Committee's standards. So for number 1 here on the naming sequence variants, you can see it was pretty good. Now let's go to number 2, reporting the variants using the HGVS nomenclature, HGVS being the Human Genome Variation Society. For this one, 0% fully fulfilled it, means that all the variants within the report complied with HGVS standards, while only 25% of them had greater than one variant that was not in compliance with it. So you can see pretty low compliance here in reporting the variants by HGVS nomenclature. Moving on to number 3, using the locus reference genomic assemblies. So remember, in the other video we talked about how this is essentially specifying which assembly you're using. 0% fully fulfilled, meaning that the reference sequence extension number and version number were reported, while only 5% partially fulfilled it, which meant that only the reference sequence extension number was reported, so not the version number. So only 1 out of the 20 companies at least partially reported it for the genes they were looking at or testing. Number 4, if available, report the DB-SNP-RS identification number. So this one should be fairly straightforward, and you would hope it would have high adherence, but only 20% of the panels evaluated fully complied with this, reporting it for all available variants. 60% at least had greater than one allele, but less than all were missing the RSIDs. So again, 60% had greater than one, but less than all were missing them. For number 5, using the haplotype translation tables, this one was very small. Only one company or panel fully fulfilled it, which was included in the partially fulfilled. So of all the 20 commercial tests, only one company fulfilled this standard and they fully fulfilled it. So both of these statistics include the same company. And so looking at that, that means all the alleles were converted according to and how they did it within their report. They specified that they did it by HGVS nomenclature. So you can see here on the naming sequence variance side, pretty low adherence amongst all of them, with very few fully adhering or fully fulfilled companies fully fulfilling each standard. Let's take a look at the test reporting itself. You would think this would be a little bit higher. And so for the first one, indicate each variant and or haplotype observed in the test report. This one, 90% of companies fully fulfilled it, meaning that they reported all observed variants and or haplotypes tested. While 100% at least partially did it, which means they reported only a portion of the observed variants and or haplotypes tested. So pretty good there. Moving on to the next one, list variants and haplotypes that can be detected by the test. For this one, only one company fully fulfilled it, while 85% of them partially fulfilled it. So what this means was the one company listed all variants and haplotypes included on the testing panel, well, 85% of companies listed variants or haplotypes included on the testing panel. So an example of this would include star alleles, but not the corresponding variant information, such as the HGVS nomenclature or RSIDs. The third standard on the reporting guidelines describe the test, including limitations, such as types of variants that cannot be detected. For this one, 45% of the companies or the panels evaluated fully fulfilled, well, 95% partially fulfilled. So what this means is that 45% of the panels describe the testing methods and limitations, while 95% describe the testing methods or the limitations. So only one company described neither. Number 4, the test description should be made publicly available on the laboratory's website or the genetic testing registry. What we can see here is that 25% of the companies fully fulfilled that, meaning that the test description was registered in the genetic testing registry or equivalent information was available on the company's website. Well, 75% partially fulfilled it, meaning that it wasn't registered in the genetic testing registry and the website provided minimal information about their test. Finally, use of CPIC consensus terms of allele function and status and phenotypes. So remember that poor metabolizer, rapid metabolizer, extensive metabolizer. Only 10% of companies fully complied with it, so they used all applicable terms complying with CPIC. Well, 90% at least used greater than one of the applicable terms, but less than all complied with the CPIC. So greater than one of the genes they reported used the terms, but not all of them. So as we can see here, when we evaluate the individual standards, we have some pretty low fully fulfilled, but maybe a little better on the partially fulfilled. So the last question is, overall, if we were to look at each of the 20 panels evaluated in this study by Boostman, what was the percentage adherence overall? So the best, fully fulfilled 50% of the 10 guidelines or standards here, but this one panel 100% partially fulfilled. So probably that company has updated now their panel, perhaps, after this study came out, and maybe they're more closer to 100% fully fulfilled. But an interesting stat is that 12 of the 20 panels included in this reported study only fully fulfilled 20% or less of the standards. So that means that a majority of the panels included in this report did a pretty bad job of fully fulfilling these standards. So perhaps there's a lot of work out there. This study is from 2017, so maybe there needs to be an updated study so we can see if these companies or these panels are doing a better job reporting their decision support tool results. So I hope this gives you an idea that there's really a wide range of reporting that can occur between decision support tools out there, just in terms of looking at adhering to CDC or CPIC advised standards, and that you should really look into each test individually to see what their decision support tool reports and if it has all the correct information or the useful information that is recommended by these expert panels. Thank you for your attention.